So looking at the heterozygous parent, that means that what are our parent total genotypes? Anybody? What are the letters? So the B's are the second trait given. So I'm going to go with the first trait's given first, and the second trait's given. Big N, little n, big B, little b. Okay, so then using your foiling out for the alleles are only, or the gametes are going to only have two alleles, one for one trait, one for another trait. So big N goes with big B. That's going to be our first gamete. Big N, big B. The second gamete is going to be the big N paired with the other allele of that trait with the eye color. So that'd be big N, little b. Then you've exhausted the combinations with big N. You have to go to the second allele for N, which is the little n. It's going to pair with big B. So little n, big B, and then the other combination would be little n with little b. And that is going to be, those are the four gametes that could be formed from that combination of alleles, heterozygous alleles. The second parent is exactly the same in terms of the big genotype, so you can make the same exact combinations for the gametes. So I'm going to write those same things here. Keep going with me. You guys can do this. All right, so then as you would normally do with a Punnett square, you're going to pull together the row and the column that are touching, except we have to fill 4 alleles in because 2 alleles are going to contribute to the first trait being black or white, and then the second 2 set of alleles is going to contribute to the other trait of blue eyes or brown eyes. So that's why this is called dihybrid, because you're considering 2 traits at the same time. So in this one, you're going to have big N, big N, big B, big B. Second one, big N, big B, little b. Big N, little n, big B, big B. Big N, little n, big B, little b. Next one, next row, big N, big B, little b. Pulling together these two, big N, little b. Next one, big N, big B, little b. Next one, big N, little b. Then we go down to the third row, big N, little n, big B, big B. Big N, big B, little b. Little n, big B, big B. Little n, big B, little b. Last row, big N, big B, little b. Next, big N, big little b. Little b. Next is little n, big B, little b, and then little n, little b, little b. Okay, let's do our color like symbols again because I don't want to write out what each of these are. What are the different combination traits we're going to be looking for? The traits, the phenotypes.
Color.
Okay, so I've got, what was the very first color given? Oh, no, I can't move it. All right, you guys have to read yours.
Normal.
What was a normal? Does it just say normal for real? Okay, so normal color, and what color eyes would be dominant? Brown. So normal and brown, we're going to give a happy face. Okay, so which of these are normal and brown? 1, 2, 3, 4, 5, 6. Nope, I lied. Thank you. Okay. 7, 9. Whatever number this is. Yes. This one? No. No. Yes. No. 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 Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9. Who knew that before we got done? Thank you. Okay, so there's 9.
Oh, yeah.
9, 3, 3. Yes, it's 9, 3, 3, 1, but I'm proving it to you, and you will have to draw out your Punnett squares. What's the next trait we're looking for? Traits. Normal and blue eyes. So we still got the dominant for the first trait, and then the recessive for the second trait. So I'm going to do a heart. Okay. So normal and blue.
Yes.
That one.
Yes.
That one.
No. No.
Yes. No. No. Okay. So 3. Next set of traits we're looking for are what? Black and then brown. Black first, and then brown for the eyes. So we'd have a recessive N and then a dominant B.
Yes, yes, yes,
Yes, yes. Ooh, no, no. Give me a different symbol. Stars. Okay. Yes, yes, yes,
no.
no. Okay, so 3, and then the last one, 1. Oh, hashtag, okay, that's you. Check hashtag, 1. And that's recessive, recessive. What's up? Okay, so I know they're long. They're not really that hard, though. It's just very tedious. Okie dokie. Quiz time, and then we'll do carry type notes in about 5 minutes, and then I'll let you guys work on your stuff. All right, so clear off your desk. All you need is a piece of paper. Go to my stash and go grab some paper. Don't be messing with people's paper accounts. Okay, so on your desk, if you have the Apple genetics, don't steal them from me. Just put them on the floor next to you or put them inside the textbook front cover, but I'll get them later. Put them out of sight and out of mind. Put them in the, I'll get them later. Thank you. I'll wait for you. Okay, after you get done with this, we're going to do the karyotype notes, but just when you're done with your quiz, just raise your hand and I'll come and grab it from you. You can see that your homework is 1 through 13 on the study guide. When I get your quiz, or maybe you can come up here and bring me your quiz actually, and just grab the study guide off my desk right here. Okay, so as we wait on everybody else to finish their quiz, I don't want to rush people too much, just grab this and start before we do our karyotype notes. On the quiz, there are 7 multiple choice questions. I don't care if it's multiple choice, I'm looking for the Punnett square. If you don't draw Punnett square automatically, if it's in your however many points, it's tap off support your answer with the Punnett square, okay? So please draw your Punnett squares. That's what we did this for warm up today. I'm going to put the dihybrid one on the board because that's the one I was going to use for the quiz. It's cool. Ready?
Yeah.
Okay. Please keep your eyes on your own paper. If I see wandering eyes, I automatically give zeros and I won't tell you to sit on Power School. That makes it right. No, please don't write on your quiz. That's why you have your own paper. To be, I'm going to show you the picture first and then I'll tell you what to write down. Karyotypes are pictures of chromosomes. Who's ever seen something like this? Anybody? Some of us have? Okay. These are a picture of all your chromosomes from one cell. So we assume that all your chromosomes from one cell is going to be the same as any other cell because your DNA is identical across all cells in your body. So what they do is they look at a karyotype to determine some sort of illness or disease that you have based on the shape and number of your chromosomes. They don't look at the little bitty, like, let me dig in there and look at the nucleotides if you have an A or a T or a C or a G. They look at the shape, size, and the number of your chromosomes. So that's what we're going to be talking about. So I just want to point that out before we actually move on. Okay, so some genetic disorders are going to be caused by having too many chromosomes. We already talked about one of them, which was Down syndrome 21. Thank you. So it's 21st chromosome that we're talking about. So I just mainly want you to remember what non-disjunction is, and that the genetic disorders we're talking about right now are springing from chromosomal abnormalities. So I say chromosomal abnormality, you can either say we got too many or too few chromosomes, or we are missing parts of chromosomes, or we have too much of a particular chromosome. So there's some differences here. And I'm going to draw out what meiosis phase we're talking about here. So we're talking about genetic disorders caused by chromosomal abnormalities. Non-disjunction is going to occur whenever you have, in meiosis 1, metaphase 1, what do the chromosomes look like? It's a good review. They're in the middle, but how are they in the middle? How are they aligned? In pairs or single file?
Wait.
Meiosis
Pairs.
1. Thank you, in pairs. So whenever you have your chromosomes lined up, they're lined up in their tetrad form, right? Well, what just happened before metaphase 1? Huh? They condensed in prophase and they formed their tetrads, but what happened in prophase 1? They were so close together. The X of the, yeah, so crossing over probably occurred right there on that tetrad, and then right here on this tetrad. So that's where we get, like, you know, the whole genetic diversity thing going on here. But when they're in metaphase 1, they're still, you know, next to their individual homologous chromosome. What happens in anaphase 1? What separates? The tetrad separates, so the homologous chromosome goes that way and that way, right? So then we end up with a cell that should have something like this. This and this. But just, you know, just FYI, if this chromosome pair does not separate appropriately, and both of those chromosomes go that way, we're going to end up in the end, when we go through meiosis 2, we're going to end up having to, you know, separate our sister chromatids, and we're going to end up with cells with not enough chromosomes and then too many chromosomes. Those cells are called your gametes, right? Those gametes then come together later on, and they're going to end up with a zygote with either too many chromosomes or too few chromosomes. So this is the problem. In anaphase 1, if this does not separate appropriately and both of them go to one side or the other side, that's the very, very beginning of the problem. And what happens in the end is we have our monosomy and trisomy, where one cell is going to be missing a chromosome in the end, and the other one's going to have too many. So what does the tri refer to here? 3. So this is, these two things are, we call them after fertilization occurs. Because after fertilization, you have 2 of each chromosome, one from the egg, one from the sperm. So if the sperm was missing a chromosome by chance, then that egg or that zygote's going to end up with a monosomy, because mono means 1 chromosome. If some other sperm had 2 of the same chromosome, while the egg only had 1, come together, and that's how you get your 3, your extra chromosome. So that's what we call trisomy. Yes?
So extra chromosome Down syndrome.
Yes. Mm-hmm. So it's called, Down syndrome is an example of trisomy 21, so it's saying the 21st chromosome, there's an extra one there. And that's really easy to see on a karyotype. You just count chromosomes. You're like, oh, wait, I got 3 on this one. I have a trisomy. Oh, I only got 1 of this one. I have a monosomy. Yeah.
So there's like trisomy, a different number. Is it still Down syndrome?
It's a different disease. And I can't think of any off the top of my head what they are called, but there's other ones like trisomy 11, trisomy 12, stuff like that, and they'll be different genetic disorders. Okay? All right, thumbs up good here? Moving on, almost in a second. Okay, so we're going to be able to see this on an image. What else can you see on an image about this individual, like on a karyotype? What else would you be able to determine about them? Okay, how big the chromosomes are comparatively to one another. What other traits about an individual?
Their gender.
Yes, their sex, their gender. You can look at the 23rd pair of chromosomes, you can see if they're the same size, they're probably both X's. But if you have one really big chromosome and one little dinky chromosome, that little dinky one is the Y chromosome, it just doesn't have as many genes on it, so that means that person is male. So that's what we're going to see here. So I'm going to, actually, I have this bigger on the next slide, I'll come right back here. On this one, you see, big, huge chromosome, little bitty chromosome, that is our male version of the karyotype. For it to be the same size would be 2 X's, that would be female. Okay? All right, let's go back here. How do they see these things? First of all, you have to know karyotype means, karyo means nucleus. So you know, like eukaryote and prokaryote, that carry kind of prefix or stem means nucleus. So what are they pulling out of the nucleus for these pictures? Type. Well, type kind of means like image of interest. Genes, all the DNA, right? So pulling out all the DNA out of the nucleus, and they're putting them in a pictorial form. Well, how do they do that? DNA is not always nice and condensed and, like, you know, take it out. It's only going to happen when your cells are in the middle of mitosis. So they find some cells that are rapidly dividing, like what? Okay, cancer cells. That would be like whole another story there. We're not looking to see like what genetic diseases you have, or like, do you have cancer? Stomach cells are ones that replace themselves every 4 days. So they might take some of your stomach cells, they'll catch them in the middle of metaphase. And when they're like kind of all lined up in the middle because they're nice and organized, they'll kind of freeze the cell so they're stuck in metaphase, and then they take a picture of it. and they take a picture of it, so they're doing this under a microscope, and they take a picture of it, they can actually look at all the 46 chromosomes you have and pair up the ones they think are chromosome 1, chromosome 2, chromosome 3, and so forth. And they can look at your similarities between your chromosomes. So I'm going to turn the lights off a little so you can see those. You guys write down what you need to there. So you may take down the things that you need to determine what these things are. So I said it was in metaphase, because that's when the chromosomes are nice and organized on the equator of the cell. They take a picture of it, and then they organize the chromosomes in pairs, because the pairs are what we're comparing. If there's any issues, you can easily tell. Yeah?
So is a karyotype just like counting the cells and finding out what's wrong?
Counting the chromosomes to figure out what's wrong, yeah. So a karyotype is a picture of what's in the nucleus, essentially. So, image of chromosomes in nucleus. And the purpose of doing this is to determine what genetic disorders this person may or may not have. Because once you put them in a picture form like that, it's really easy to look and see where the differences lie. So what's the difference you automatically see between some chromosomes? The very first pair, that's chromosome pair 1, chrome pair 2, chrome pair 3, chrome pair 4, blah, 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 chrome pair 23. What is the difference you see in that picture? Andrew?
Well, for 2, the left chromosome is shorter and it's like curved.
Okay, so it's kind of curved. That might just be how like maybe it caught it right after metaphase was happening, the chromosome was starting to be pulled apart. In anaphase, they kind of have that kind of a V shape. But you also can't really tell, but that might be a deletion of the chromosome right there. That might be an issue. What else do you see? Another chromosome? I see that one's kind of the same. It's kind of got that little shape to it. Anything else that's vastly different? Gavin?
The middle one that's circled has like 3 different colors.
Oh, that one's got like 2 different colors. So that's really different. We're going to see what that is in a second. Yeah.
All the pairs are different sizes.
Okay, yeah. So you'll notice that a lot of times it kind of goes from biggest pair all the way down to smallest pair. So karyotypes are very well organized, so that and the geneticists can actually determine a lot about the person's chromosomes. So they commonly call chromosome 1 the biggest pair and the chromosome 22 the smallest pair. All right, so let's look at some of the mistakes and differences here. Oops, made that really big. Okie dokie. Oopsies. I don't know what's going on here. Okay. You see on this chromosome here, what do you just shout out? What do you observe?
Smaller.
Smaller. So part of it's deleted. That's called a deletion. They were really creative in making that word. It's called a chromosomal deletion. This pink part here, where do you think it came from?
Pink part on the left.
The pink part over here. So this was deleted from this chromosome, and inserted right there. So guess what this type of abnormality is called? An insertion. Okay? Or an addition, sometimes it's called an insertion. So where do you think this came from?
The other one that's circled.
Which other one? Oh, so this deletion down here, and it was translocated over here. Okay? So I'm saying the word insertion and translocation. This is an insertion from another chromosome. This is also called a translocation, because what does the word trans mean? To move. Location means to move location, so translocation moves location. It's moving it to another chromosome. Those are all genetic defects and genetic disorders we're going to be having to identify from a picture, or it might just give you the definition. So, because I think these are really pretty. Chromosomes are not that color. They paint them like with Photoshop to illustrate the differences between the chromosomes. So that is one thing, you're not going to see that in your cells. That'd be awesome, but you're not that cool. Okay, I'm going to let you guys write down this, and I'll come back to that. So I'll skip this for a second. These are the types of abnormalities we were just discussing based on the pictures of the karyotypes. You guys can write down at least the golden words. If you get it, you get it. Because you're like, duh, I know what a deletion is. Yep, sure, I know what a duplication is too, but at least write down those. So deletion is obviously when something is just deleted and it's gone. So we have like a chromosome, and then we have the little bitty one, and so this part right here was deleted. A duplication event is where parts of the DNA are duplicated, just a portion of it though. So this was our deleted. Duplication might be on my chromosome. I had blue, white, blue, white, and so this is just painted. But to duplicate that, I would just really double the imagery here. And then secondly, blue, white, blue, white, blue, white. So duplication is just a duplication of the DNA. Translocation, like I said, it's transported, so move. Location is what translocation means. And it's going to be inserted on another chromosome location. Inversion. Take your pencil like this and invert it. You just flip it. So you have your pencil upright. Inversion is just flipping. So if I had a DNA segment and I actually could read the DNA and it was ATGC and it was this way on the chromosome, invert that. CGTA, right. So an inversion is just flipping taking it out of the chromosome, flipping it, and putting it back in. And that's within its own chromosome. It's not inverting it and then like translating it or translocating it to someplace else. Scrolling up a little bit here. Can I go further? Anybody? Holler if it's not good. You can see if I didn't have these labeled, you could see the differences. You can see that that part is doubled and extends the length of that chromosome. You can see this part, the fat yellow part's on the top and then the fat yellow part's kind of ish on the bottom here, but it's inverted. You can see that totally gone, it's deleted. You can see the insertion. This chromosome purple part is inserted in the middle of the black region to extend the black region. And so there would have to be some like markings on there for you to really determine it. But if I gave you pictures, could you think you could determine which chromosomal abnormalities existed? Yes? Yeah?
Yeah.
Yeah, thank you. And then translocation, nothing is cool. So all of these were called chromosomal abnormalities based on shape. Okay? So which ones did we talk about that were based on number? Trisomy and monosomy. And the ones that are based on shape are deletion, inversion, duplication, blah, blah, blah. Okay? All right. Can I get one volunteer? We are going to experience carrier shop. Like Photoshop, but way cooler. Carrier shop. You ready? Gavin's my chosen one. You are the chosen one. This is not, well, here, actually, let me show you this first.
The chromosomes are first separated from the background by adjusting a threshold. Notice the outlines expand and contract.
So they take
The default
pictures.
setting lets the software choose the settings, and the aggression level determines how aggressively chromosomes are split apart. The next stage is to cut up the chromosomes that are still touching. This can be done manually like this, or in this case, a double click on the mouse allows the software to separate the chromosomes for you.
They literally have carrier shop. This is not fake.
Overlaps can be resolved automatically with a double click. Here, these two are separated. Or in this one, we'll use a manual tool to paint across the chromosomes, and the software will separate these and resolve the overlap. Here's another overlap.
So geneticists do this if they're trying to determine something about like an unborn baby, like some sort of genetic defect.
Now we have 45 chromosomes. A gallery view lets you see all the karyotypes belonging to a case, and the composite view lets you see multiple karyotypes grouped by chromosome. SmartType runs on Apple Macintosh and on Windows PC. It's available as a software.
So you can actually buy that and arrange your own karyotype, which I thought was kind of cool. Like, kind of cool, people? Come on. I have another one for you, but where did I put it? That is the question. I may have, hold on. Pause. I've hidden it. Here it is. All right, Gavin. It's all you, man. Can you pair? Can you pair at least two of these? Get up and do it. Let's go. You guys have to holler and help him. These are the 1 through 22 or 23 already made, and he's gonna pair them up.
How do I?
You just drag. Drag it over. Come on. Go, Gavin. Go. Drag one.
Number 19. Do number 1.
No, you have to drag from the mess here to
Do number
the
1.
karyotype.
The longest one.
The longest one, yeah. So you start the longest.
No,
And-
it's not.
Yeah.
We'll drag it a little bit to the right of it and drop it. Oh, is it number 1? No, apparently it's not. So is it number 2?
Get the one that's on
This one's number 2?
Maybe. Let's see.
No.
Oh yeah. Okay. Where's number 1, guys?
The one down there.
On the bottom. It's got a little light on the top.
Down.
Yeah. right, let's do one more for kicks and giggles. I see it. You guys see it?
Yeah,
Ready?
it's all three, I'll see it.
That's 3.
Yes! See, once they're all cut apart, it's easy, but cutting them apart when they're all laid up on top of each other is kind of rough. Yay, Gavin, thank you. Okay, so you can actually do this yourself, too. All right, I'm going to hold off on test crosses and Halloween karyotypes. I'm going to let you guys work. We got about 40-ish minutes left. Remember, you want a visual to go along with your presentation, to be an intriguing presentation, so someone's like, yeah, I want to fund that research because I want to discover the cure to vampirism or something, okay? So you want to make sure that you are really promoting your case in a very good way. Know the answers to all your questions, though, because I'm a question asker, okay? All right, work hard, play hard.
I
Yeah.
did a song for our